Hello children, today we will learn chapter 3, Human Reproduction. Humans are sexually reproducing and viviparous, meaning they give rise to young ones. The reproductive events in humans include formation of gametes, which you call as gametogenesis, that is, sperms are produced by the male and ovum in females, transfer of sperms into the female genital tract, which you call as insemination, and fusion of male and female gametes called as fertilization. This ultimately leads to formation of zygote. So the fertilized egg will develop as a zygote. This is followed by formation and development of a blastocyst. What is a blastocyst? It's the early stage of an embryo, which is a cluster of cells which are capable of dividing. So this is made by a fertilized egg. So this is how an embryo develops. And embryonic development is called as gestation, and ultimately it ends with delivery of the baby, which you call as PART-1. Yes, children? So this is an introduction to sexual reproduction in man. Now we will see about the structure of the male reproductive system. So the male reproductive system is located in the pelvic region. It includes a pair of testes along with accessory ducts, glands, and the external genitalia. So what are the structures included in the male reproductive system? You got a pair of testes along with external genitalia, which you call as the penis. The male accessory ducts. What are the different ducts? Rete testis, vas efferentia, epididymis, and vas deferens. It also includes the male accessory glands. So you have got ducts, you have got glands apart from testes. So what are the glands? They are the prostate, the seminal vesicle, and bulbourethral glands. So the external part of the male reproductive system includes the penis, the scrotum and testicles, and the others. The internal parts include vas deferens, ejaculatory ducts, urethra, prostate gland, the bulbourethral gland. Of all these, the penis and urethra, they are a part of both urinary system and the reproductive system. So we call that as the urinogenital system. The penis and urethra, they are a part of both systems, the excretory system, urinary system, and the reproductive system. So you call it as urinogenital system. Now let us learn something more about the testes. The testes are situated outside the abdominal cavity within a pouch. What do you call that pouch? It is called as scrotum. So a pair of testes situated outside the abdominal cavity within a pouch called scrotum. The scrotum helps in maintaining the low temperature of testes, that is around 2 to 2.5 degrees Celsius, lower than the normal internal temperature. So what is scrotum? Scrotum is a sac of thick skin that protects the surrounding testes, and it helps in maintaining low temperature of the testes around 2 to 2.5 degrees Celsius, lower than the normal internal temperature, that is lower than the temperature of the body. So the normal body temperature is around 97 and 99 degrees Fahrenheit or 36 and 37 degrees Celsius. Around 2 to 2.5 degrees less than this is the temperature maintained in the scrotum, as it acts as a protective covering for the testes. So the testes are covered by 3 protective coverings, which are called as tunicae. They are externally covered by 3 protective layers: tunica vaginalis, tunica albigine, and the innermost layer, which you call as tunica vasculosa. So in adults, each testis is oval in shape with a length of about 4 to 5 centimeters and a width of around 2 to 3 centimeters. and each testis has about 250 compartments, which you call as the testicular lobules. So how many compartments are, how many testicular lobules you have in each testis? 250. So we learned that there are a pair of testes. So in male, how many testicular lobules you will have? 500 lobules, because each testis has 250 testicular lobules. So now let us see what these lobules are. So each lobule contains 1 to 3 highly coiled structures, which you call as the seminiferous tubules. So how many seminiferous tubules are present in each lobule? Around 1 to 3. They are coiled structures. These highly coiled structures are called as the seminiferous tubules. So what is the function? Because it is the place where the sperms are produced. So the testicular lobules are small compartments that contain highly coiled seminiferous tubules, which are ranging from 1 to 3. In each lobule contains 1 to 3 coiled seminiferous tubules where the sperms are produced. So each seminiferous tubule is lined on its inside by two types of cells. So the inner lining of seminiferous tubule is made up of two kinds of cells, two types of cells. What are they? They are the male germ cell, which you also call to be spermatogonia. The male germ cell is also known as spermatogonia, and the other kind of cell is Sertoli cells. So what are the two kinds of cells that are present in the inner lining of the seminiferous tubule? They are the male germ cell, which is otherwise known as the spermatogonia, and the Sertoli cells. So what is the function of the spermatogonia? Spermatogonia go through spermatogenesis to develop into mature spermatozoa. So by the process of spermatogenesis, this spermatogonia produces mature spermatozoa. So the male germ cells undergo meiotic divisions leading to sperm formation. And what is the function of the Sertoli cells? They provide nutrition to the developing germ cells. Let us learn something more about that. Sertoli cells. These are somatic cells. What do you mean by somatic cells? These are vegetative cells in the testes that are essential for spermatogenesis. They are also known as nurse cells. You call these as nurse cells because they provide nutrition to the germ cells. Yes, and there are one other kind of cells called as the Leydig cells or the interstitial cells. So what do you mean by these interstitial cells or Leydig cells? These are the cells which are located in the testes between the seminiferous tubules. That is, the region outside the seminiferous tubule is called as the interstitial space. So the region outside or in between seminiferous tubule, you got some region, isn't it? That region is called as the interstitial space. And what do you find in this interstitial space? You have some blood vessels and the Leydig cells. You have some blood vessels and the Leydig cells. So what is the function of Leydig cells? They synthesize and they secrete a testicular hormone called androgen. One important hormone of androgen is testosterone. This is the male sexual hormone, and androgen is responsible for the development of secondary sexual characters in male. So is it clear, children? So we learnt about Sertoli cells, the interstitial cells, and Sertoli cells are known as the nurse cells as they provide nutrition to the germ cells, and the interstitial cells or the Leydig cells, they synthesize, they produce, they secrete androgen, and one important androgen is a hormone, that is testosterone, which is a sex hormone, and this is responsible for producing secondary sexual characters in male. So hope you understood. So here is a concept map. So the male reproductive system, it has the male accessory ducts, the testes and male accessory glands. So three different structures are present. They are the male accessory ducts, the testes and male accessory glands. So what are the male accessory ducts? The rete testis, vasa efferentia, epididymis, vas deferens. Rete testis, vasa efferentia, epididymis, vas deferens. These are the four different kinds of ducts. Next comes the organ testis, which is the primary organ, which is protected and kept in the scrotum. Next is the male accessory gland. What are glands? Glands are organs which produce certain chemicals. They produce hormones and which are carried to different, which are transported through ducts. So here we have three different kinds of hormone, accessory glands, the seminal vesicles, the prostate gland, and bulbourethral gland. What are the three different accessory glands present in the male reproductive system? Seminal, vesicle, prostate gland, bulbourethral gland. Yes, and these male accessory ducts, you find the testicular locule which has got 250 compartments in each testis, which is nothing but the seminiferous, it possesses seminiferous tubules 1 to 3, and the seminiferous tubule produce sperms by that is the spermatogonia forms sperms by meiosis, and they also have Sertoli cells which provide nutrition, and the Leydig cells secrete androgen which secretes a hormone called testosterone, that helps in producing male secondary sexual characters in male. So this is the male reproductive system structure where you find the bladder. You got the three different glands. The seminal vesicle, this is the prostate, and these small structures, the bulbourethral gland. And you got the other ducts, the epididymis and the vas deferens. This is the structure of the male reproductive system. Yes. So now we look into the process of spermatogenesis. Spermatogenesis. What is spermatogenesis? How do you define spermatogenesis? It is a process by which haploid spermatozoa. Spermatozoa, nothing but the sperms. What is the ploidy? They are called as haploid structures. Why? It carries one set of chromosomes developed from the germ cells. That is the spermatogonia or the sperm mother cell. You call this sperm mother cell or spermatogonia. And where are they present? In the seminiferous tubules of the testicles. We learnt about this, isn't it? So let me repeat the definition of spermatogenesis. It is a process by which haploid spermatozoa develop from spermatogonia or germ cells in the seminiferous tubules of the testicle. Yes. So what are the different steps involved during spermatogenesis? So during development, the primordial germ cells, they migrate into the testes and they become immature germ cells called the sperm mother cells. So initially, what is formed first? The spermatogonia or sperm mother cells are formed in the inner surfaces of the seminiferous tubule. And what is the ploidy of this spermatogonia? It is diploid. So a diploid spermatogonium, that is the male germ cell, first increases in size to form a diploid primary spermatophyte. Right? This diploid primary spermatophyte undergoes meiotic division. So what is the first division that takes place in the primary spermatocyte? It undergoes meiosis 1. You call this meiosis as reduction division. Primary spermatophyte is diploid, and as a result of meiotic division, which you call as reduction division, they produce two haploid secondary spermatocytes. So primary spermatocytes are diploid and secondary spermatocytes are haploid. First, the process begins with the spermatogonia, which undergoes mitosis and produces primary spermatocytes, diploid primary spermatocytes. These diploid primary spermatocytes further undergo meiotic division, which you call as meiosis 1, that is reduction division, where the diploid primary spermatocyte produces two haploid secondary spermatocytes. So the ploidy of primary spermatocyte and spermatogonia, they are diploid, whereas secondary spermatocytes are haploid. Now, each secondary spermatocyte further undergoes a second meiotic division. This is called as meiosis 2. And it forms two haploid spermatids. These secondary spermatocytes further they undergo a second meiotic division. Yes, the secondary spermatocytes are haploid. Is it clear? They are haploid. They again undergo a meiotic division and they further produce two haploid spermatids. So what is the difference between mitosis and meiosis 2 is mitosis happens in a diploid cell, whereas meiosis 2 it happens in a haploid cell. So a secondary spermatocyte which is haploid undergoes a second meiotic division to produce two haploid spermatids. So how many spermatids are formed? 4 haploid spermatids are produced from a diploid spermatogonium. So from one diploid spermatogonium, we get 4 haploid spermatids. These spermatids are transformed into spermatozoa by the process called spermiogenesis. This is the last process where the spermatids are transformed into spermatozoa. And there's one other process which you call as spermiation. What is this spermiation? It is the process by which mature spermatids. See, after the formation of these spermatids, mature spermatids are released from the supporting somatic cells into the lumen of the seminiferous tubule. So this process of release of mature spermatids into the lumen of the seminiferous tubule is called as spermiation. So you need to remember all these. You should know to define what spermatogenesis, what is spermiogenesis, and what is spermiation. Okay, what is spermiogenesis? That is the spermatids being transformed into spermatozoa. That process is called as spermiogenesis. Spermiation process by which these mature spermatids are released into the lumen of the seminiferous tubule. So this is all about spermatogenesis. See, this is a schematic representation. See here, spermatogonia is 2N. 2N is diploid. It undergoes a first mitotic division producing two primary spermatocytes. Yes, each spermatocyte is again diploid because it undergoes only a mitotic division. So the ploidy will not change. Whatever the mother cell has, the same ploidy will be present in the daughter cell also. So spermatogonia is diploid. So it produces two diploid primary spermatocytes. So each diploid primary spermatocyte undergoes meiosis 1, reduction division, producing two daughter cells which are haploid in nature. And how many? Two they produce. So you call this as secondary spermatocyte. So primary spermatocyte is diploid, secondary spermatocyte is haploid. This secondary spermatocyte again undergoes a division meiosis 2. This is reduction division, but this is not reduction. Meiosis 1 is reduction division. Meiosis 2, where each secondary spermatocyte produces two spermatids. So as two secondary spermatocytes are there, you get 4 spermatids produced from one spermatogonia. One diploid spermatogonia produces 4 haploid spermatids, and they, by the process of spermiogenesis, produce spermatozoa. This is the schematic representation of spermatogenesis. See, I just represented this here again. Spermatogonium. Yes, diploid. One spermatogonium. First it undergoes mitosis. So produces two primary spermatocytes. Each primary spermatocyte, again that is diploid, undergoes meiosis 1 and produces two secondary spermatocytes. Each secondary spermatocyte, which is haploid, undergoes meiosis II and produces 4 spermatids. All these 4 spermatids later mature as spermatozoa or the sperms. Is it clear, children? Hope you understood. I have repeated it many times, so by this time you should be thorough in this. And this is the sectional view of the seminiferous tubule. So these are the seminiferous tubules, and this is the interstitial space which you find between the spermatogonia. Each one is a spermatogonia, and between spermatogonia you find some space, isn't it? You call that space as the interstitial space, where you have the interstitial cells or the Leydig cells. And these are the spermatozoa and the Sertoli cells, which you find on the inner lining of the on the seminiferous tubule you find the Sertoli cells and the spermatogonia. This is an enlarged section of the seminiferous tubule. See, such questions may come in the examination, where you will be asked to label all these parts. So what is this part A? This is spermatogonium. This is the spermatogonium. See, one portion alone is being enlarged. A section of this is enlarged here. Yes, so this is the spermatogonium. And what is B? B is the Sertoli cells. B is the Sertoli cells, and C is primary spermatocyte. These are primary spermatocyte, and D is secondary spermatocyte. D is secondary spermatocyte, and this one bigger one, blue colored structure is seen, isn't it? That is a spermatid, and these small structures, two blue color structures, they are spermatozoa or the sperm cells. Is it clear, children? This is a portion of the seminiferous tubule that is being enlarged. So such questions may arise in the examination. They may give you this structure and ask you to label. I think it's clear now. Now let us see the difference between spermatogenesis and spermiogenesis. Difference between spermatogenesis and spermiogenesis. Spermatogenesis is the formation of motile spermatozoa from spermatogonia, and spermiogenesis is the formation of mature spermatozoa from spermatids. So here from spermatogonium, the process that includes all the events that include the formation of spermatozoa from spermatogonium. Here it is the maturation, that is the formation of mature spermatozoa from spermatids. 4 functional spermatozoa are produced from a spermatogonium. This we saw earlier. One spermatogonium, diploid spermatogonium, produces 4 haploid spermatozoa. Here, one single spermatozoon is produced from one spermatid. Each spermatid transforms into a spermatozoon. Yes, and so in spermatogenesis, it consists of a multiplication phase, growth phase, and maturation phase, and a differentiation phase. Here, it consists only of a differentiation process. Yes, where the spermatids differentiate as spermatozoons, and diploid germ cells become haploid because spermatogonium is diploid, and spermatids, the spermatozoa, are haploid. This occurs, spermiogenesis occurs in haploid cells. Thus, there is no change in the amount of genetic material. This is the difference between spermatogenesis and spermiogenesis. Hope it is clear, children. Next we'll see something more about the glands, the male accessory glands. The accessory glands, seminiferous tubules, they open into vasa efferentia through rete testes, and vasa efferentia leaves testes and open into epididymis on posterior of each testis, which further leads to vasa deferens that ascends to the abdomen. So this is the connection between the ducts. And urinary bladder receives duct from the seminal vesicle also, and it opens into the urethra, which is nothing but the which opens as the ejaculatory duct, where the sperms are stored from the testes. So what is urethra? It is a tube-like structure that extends from the bladder, and it continues throughout the penis, and it opens at the end, the external opening of the penis called the urethral meatus. And this is the line of connection, how the contents are transferred from the seminiferous tubule to retate testis to vasa efferentia, epididymis, vas deferens. Ultimately, it reaches the ejaculatory duct, where you have a duct from the seminal vesicle also. So next we'll be seeing about the glands. There are a paired seminal vesicle, prostate and bulbo-urethral glands. All these, they produce seminal plasma. What is the seminal plasma? What are the secretions of these glands called as? The secretions of these glands are known as seminal plasma. So what is seminal plasma? It is the chemical substance that is being secreted by all the three glands. And what is the function? They are rich in fructose, calcium and certain enzymes, and secretions of the bulbourethral helps in the lubrication of the penis. This is about the male accessory glands. Yes, I hope you understood, children. I'll just repeat it once again. The male sex accessory ducts include retate testis, vasa efferentia, epididymis, and vas deferens, and the seminiferous tubules of the testis open into vasa efferentia through retate testis. And the vasa efferentia leaves the testis and opens into epididymis located along the posterior surface of each cell. So all these ducts store and transport the sperms from the testes to the outside through urethra, through the opening called as urethral meatus. So, and the penis, which is the male external genitalia, is made up of a special kind of tissue that helps in the erection of the penis to facilitate insemination. Yes, and the enlarged end of the penis is called the glans penis, which is covered by a loose fold of skin called as the foreskin. And finally, the secretions of the vulvourethral gland, they also help in the lubrication of the penis. With this, this portion gets completed. Next, we'll see some questions. That's our question on assertion and reasoning. Assertion is primary spermatocytes of testes are haploid. Primary spermatocytes are formed by meiosis 1 in spermatogonia. See, the options given, both assertion A and reason are true, and the reason is the correct explanation of assertion. This is wrong. Why? Because primary spermatocytes are not haploid, they are diploid. And the second reason, primary spermatocytes are formed by meiosis 1 in the spermatogonia. So that both the statements, both assertion and the reason are false. Yes, because primary spermatocytes are formed by mitosis in the spermatogonia, not by meiosis. Is it clear? So both these statements are false. So answer is D. Both assertion and reason are false. There's one other question. Where are the Leydig cells present? What is the role in reproduction? Leydig cells are present in the region outside the seminiferous tubules called the interstitial space. Why are the human testes located outside the abdominal cavity? Name the pouch in which they are present. Testes are located outside the abdominal cavity to maintain a low temperature of testis, which is necessary for spermatogonium. And where are they? Name the pouch. Pouch is scrotum. The next question, write the location and function of the following in human testes, Sertoli cells and Leydig cells. Where is the location of the Sertoli cells? There it is, in the lining inside the seminiferous tubules. What is the function of Sertoli cells? They provide nutrition to the germ cells. And what is the position of the Leydig cells? They are outside seminiferous tubules in the interstitial space. Yes, and what is the function? They synthesize and secrete androgens. Yes. With this, our study gets completed. Hope you all understood, children. Thank you.